Hello and welcome to our presentation on the TCA cycle, also known as the Krebs cycle, by Henry Fisher, Dawson Bryan, and Caleb Murphy of Group 61. What is the TCA cycle, which is also known and referred to as the Krebs cycle? The TCA cycle is a pathway that is used to generate energy and build amino acids by the oxidation of acetyl-CoA. Although there are more purposes, those are just to name two. But this cycle comes right after the aerobic glycolysis, where glucose breaks down into pyruvates, and then those pyruvates into the cycle after they are converted into acetyl-CoA or CoA. Real quick, before we can talk about each step, this is the overall pathway of the TCA cycle. In total, there are 9 steps, although technically there's 8 for the presentation of this TCA cycle. There is 9. But before we can start with step 1, we must understand that acetyl-CoA must enter. In the first step of the TCA cycle, oxoalloacetate reacts with a water molecule and acetyl-CoA to produce citrate, a 6-carbon molecule. This step is exergonic and is a highly important step within the Krebs cycle. Also, during this step or reaction, the CoA molecule is kicked off, or the CoA molecule is kicked off. This step is so important that sometimes the TCA or Krebs cycle is referred to as a citric acid cycle. Although this photograph is very jumbled, we can see, or what's important is that the water in the bottom in the acetyl-CoA along with the oxoalloacetate come together in the cycle to form citrate. In step 2 of this reaction, the citrate molecule that we just formed reacts with aconitase to form the cis version of aconitase, or nactate. However, in this reaction, H2O is an off product. In step 3 of this reaction, the cis actonitate that we just formed reacts with actonitase again in water to form isocitrate. In step 4 of this reaction, the isocitrate that was formed reacts with isocitrate dehydrogenase with an off product of CO2 and NADH to form A-ketoglutarate. A-ketoglutarate is also alpha ketoglutarate. The NADH comes from the molecule of NAD being reduced, while the structure now only has 5 carbon molecules. In step 5, the alpha ketoglutarate or A-ketoglutarate reacts with the A-ketoglutarate or alpha ketoglutarate dehydrogenase complex and the CoA or CoA to form succinyl-CoA or CoA. In this reaction, the NADH and CO2 are the off products. This process causes the molecule to turn into a 4-carbon molecule. In step 6, the succinyl-CoA or CoA reacts with succinyl-CoA synthase enzyme in a singular molecule of both GDP and a phosphate to make succinate. A molecule of GDP and CoA or CoA are off products of this reaction. In step 7 of the TCA cycle, succinate reacts with succinate dehydrogenase enzyme to make fumarate. The off product of this is FADH2. In step 8 of the TCA cycle, fumarate reacts with H2O and fumarase to make L-malate. Although in the photo it has malate, for us it's L-malate. In step 9 of the TCA cycle, L-malate reacts with malate dehydrogenase to make oxoalloacetate. The off product of this is NADH by NAD being reduced. The reason this process is called the cycle is because that same oxoalloacetate that we just formed is then reacted again with step 1, which we talked about previously. Once it reacts with the acetyl-CoA water, it forms citrate and the cycle continues to go on. And that is the end of our presentation. I hope you enjoyed